In this video, we will cover the integration with single-cell RNA sequencing reference data step when analyzing Visium spatial transcriptomics data in Chipster. In this step, we can predict the cell type composition in each spot by integrating with a single-cell RNA sequencing reference. We have two tools for this step in Chipster. The first tool normalizes and runs dimensionality reduction for the reference data, after which the spatial data is integrated with the single-cell reference using anchor-based integration. The second tool visualizes the predicted underlying composition of cell types in each spatial spot after integration. Remember that we have a small number of cells in each spot, as can also be seen in the figure here. We want to predict the cell types in each spot using an annotated single-cell reference dataset. The reference data should contain all of the expected cell types, ideally from the exact same tissue. In this step, we first normalize the reference using SC transform, and then we run PCA on the reference data. We also used UMAP to visualize the reference dataset. We then apply anchor-based integration to transfer the annotations from the single-cell RNA sequencing reference to the spatial data. This integration is similar to the integration method introduced in the single-cell RNA sequencing data analysis tutorial videos, except we do not correct or modify the gene expression data in the spatial dataset. And instead of learning a joint structure, we project the PCA structure of the reference onto the spatial data. After integration, we can use another tool to visualize the predicted underlying composition of cell types in each spatial spot. Now let's take a look at how to use these tools in Chipster. We can run the integration using the Sura object after the clustering step, or we can also use the subsetted object for this step. If we use the subsetted object, we first need to renormalize the object and run PCA using this tool. We choose the parameter values here and run the tool. We can then use this tool to integrate with the single-cell reference dataset. We first need to upload the reference dataset into Chipster and then select both the new spatial object and the reference. We can then choose the number of variable genes to return an SC transform for the reference dataset. We can also choose to lower the number of subsampling cells in SC transform to speed up the computation. We also need to choose the number of PCs to compute when running PCA on the reference data, as well as the number of PCs used for running UMAP on the reference. We also need to select the number of PCs used for performing the integration here. And now we can run the tool. This tool outputs the integrated Sura object as well as a UMAP of the reference dataset. We can then select the Sura object and use this tool to visualize the integration results. We can choose which cell types to plot and also choose parameters for identifying and visualizing spatially variable cell types, similarly as we have done before. We can then run the tool. Finally, here are the results for the integration with a single-cell reference data step. Here are the predicted percentages of the selected cell type in each spot, and here are visualizations of the top spatially variable cell types.